This is episode 83 of the Immunology Podcast, Biomolecular Sensors with Dr. Shana Kelley. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Roud. Welcome back to the Immunology Podcast, where we have conversations with immunologists. The Immunology Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you enjoy the Immunology Podcast, rate us and leave a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today we have Dr. Shana Kelley from the Chan Zuckerberg Biohub Chicago on the podcast to talk about her research in biomolecular sensors and inflammatory disorders. We also got our usual roundup of recent highlights in immunology news coming up. But first,
are you studying immune cells? Get a free infographic wall chart that provides an overview of key cytokines for expansion, differentiation, and characterization of major immune cell types. To learn more, visit stemcell.com/cytokines-wall chart.
All right, Brenda, have you voted yet for us?
What are we voting for?
Well, we, Brenda, have been nominated for Best in Science and People's Choice at podcastawards.com. So if any of us, our listeners, want to go to podcastawards.com and vote for us, please do so. Voting closes July 30th, and we like to win things. It's been a busy week. It was such a busy week. Some of the stuff that came out like Tuesday and Wednesday of before this recording, we couldn't get to. So you may see that on the next round. But just in brief immunology news not being covered, there's a new magical humanized mouse coming out. And then there was also a big lupus paper that made the rounds on how interferon seems to be a master regulator of lupus that came out in Nature. But I haven't had time to review all of those because those came out in the post getting ready to review things scrambled. But keep your eyes out for some coverage of that in the future too. But in other news of busy science time, we have a paper in Nature called Inhibition of M. tuberculosis and Human ATP Synthase by BDQ and TBAJ587.
I am dizzy.
That is a lot of letters. So the first authors is Yu Ying Zhang and Zuizheng Lia, and last author is Hongri Gong, and then the second last author and corresponding authors are Yan Gao, Fengjiang Liu, and Ji Rao.
The Immunology Podcast, butchering names since, 2021. We're sorry, dear listeners. We try. We really do try.
So this is a brief paper. Why is it a big deal? Why are we talking about tuberculosis? Well, TB is a problem still, especially in parts of the world, other parts of the world, and you get resistance as well to various drugs. One of the newer drugs is bedaquiline, BDQ. It's a diarylquinolone, which was developed in M. smegmatis, and it is an ATP synthase inhibitor. Well, it has a couple of problems. One, it's really lipophilic, which means it has a long half-life, which promotes resistance. Two, it is having rapid resistance develop. And three, it inhibits human ATPase, leading to toxicity, or ATP synthase, sorry. Well, why didn't they fix that? Well, there's another drug, TBAJ587, that's an investigational compound. It's known to also be an ATP synthase inhibitor. Seems to affect less the human, less lipophilic, so it has better biodistribution. So that's great. But this has all just been done by screening against M. smegmatis, because guess what they don't have at all is a crystal structure of M. tuberculosis's ATP synthase. They have no structure. So this group didn't use X-ray crystallography. They used cryo-EM and got a resolution of 2.8 angstroms and got a cryo-EM structure of ATP synthase of M. tuberculosis with the drug bound, both of these drugs bound, and compared it to human structure of human ATP synthase, and thus we're able to crack the code on how these drugs are binding, where they're binding, and how you'd want to, how the differences between BDQ and TBA, right? TBAJ, because there's some, they're structurally different, and being able to see both of these drugs, which have known differences, both of the human ATP synthase and the tuberculosis one, now, after this paper is published, will enable better molecular targeting, because this is a great class they want to go after, but they've just been doing it completely blind because they don't know how the drug interacts with the structure. And so this is really an infectious disease paper, but it's also a structure function paper. And as we even saw in, when we talked about from a antibody level, but also has been discussed in, small molecules, you can now use AI and predictive algorithms to try to figure out how to make better drugs. But you can't do that if you don't know the shape or the structure of what you're drugging at all, and if it's a big mystery. And so they've been able to crack that mystery, and that's why it's in Nature. There's not as many structures that get into Nature these days, but this is a really hard one to get after, and it's a primary structure of a druggable target in an infectious disease, and that's a really big deal for them to get it. And then they do some structure function stuff in here and say, hey, look at the inhibition and how these compare. And that but it's all about the structure, and they got there with really good resolution. So that's what this is about. It's if you're A in this field, that's great. B, if you're like, I need structure of things and it's becoming a problem, I recommend people go look out to cryo-EM. I even started exploring it when I was a postdoc as a tool that you could use, and it's not like the 8-year project that sometimes X-ray crystallography has been. It is much more accessible to people who need a structure component added to their work than previous technologies, and this kind of shows that. It was very cool.
Maybe I missed it. So they're studying the structure, and are there any useful implications for therapy when it comes to like designing new drugs? So we understand better.
Well, right. So now they know where in the protein the drug binds to block its mechanism of action. And they know how resistance is being developed and where the mutations are, and how the resistance for the first drug doesn't occur with the second drug because of how it binds differently and causes a structural change that's slightly different. And they can then see as well how the differences in human ATP synthase structure from tuberculosis make it so that the second better drug doesn't inhibit in the same way. And so they're now able to pinpoint the exact residues. And if you know the residues that are important for the function, you can then change the drug through different moieties to either interact or not interact or bend it differently to cause what you want without screwing up the other one. So they have the whole suite, right? They have, pathogen structure, human structure with both drugs, with the differences in biology between them. And that's enough now, because they have structures with the drugs bound as well to be able to start doing the work to do the molecular medicinal chemistry or AI-based chemistry as well, right? And crank out new drugs. But you couldn't do that until they did this. There was just random screens and hope what you get.
Could they use AI for this?
Yes. That is kind of what I would assume they would do. But you can't feed the AI nothing. You have to, data, right?
Right. I mean, anything that is meant to treat tuberculosis is super important. There's still so many people dying of it, right? Which you would think. I think we forget how prevalent TB is across the world, and how much of a burden it is to so many people. So kudos to the authors and to the researchers involved in any kind of tuberculosis research. All right, let's talk about innate immunity, which is not my forte. So I hope I say this right. But this paper I'm going to talk about is called Molecular Definition of the Endogenous Toll-like Receptor Signaling Pathways. And it's also, talking about structure, maybe not as minute as using cryo-EM, but they do some sophisticated microscopy and things like that. First author, Daniel Fish, from the lab of Jonathan Kagan at Harvard Medical School. And this was published also in Nature on July 3rd. And as I say, innate immunity, not my specialty. I kind of tend to ignore, all these PAMs. Jason is shaking his head. It just pains him to hear that. But I know it's important, whatever. And I know that there's still not a lot of there's still some uncertainty about how, one of the workhorses of innate immunity works, which is toll-like receptors and how the signal from these receptors makes its way to the nucleus and to do something, right? So toll-like receptors are, of course, these proteins that are mostly surface proteins. You also have some that are endogenous or they're inside the cytoplasm, but basically they can detect all these different molecular patterns and activate particularly innate immune cells to initiate an immune response. And for what I understand, I was thinking of my textbook understanding of toll receptor. There is this one protein that is very important in kind of translating and kind of translating this signal, and it's called it's called an adapter protein. It's part of like making the toll-like receptor complex work. It is necessary for toll-like receptor signaling. If you don't have it, then you don't have it. There's no signaling. And here, Jason, I have a question. How do you call this protein? How, you know which one I mean? The adapter protein?
ID88.
Mighty 88. Okay. Because I have heard different ways of pronouncing it. I said MYD 88. At least that's how my lab said it back in the day. I also heard people say mid 88. But I think my D88's probably it's a little bit of a tongue twister for if you ask me. Anyway, so my D88 associates itself with the TLRs and also associates with other proteins, particularly this IRAC proteins, and this then initiates downstream all the signaling, all this activation of this pathway. But let's focus on this part because there is MID88. It's not just kind of floating around in the cytoplasm. But what this paper kind of really shows, and I think it builds on previous knowledge, so it's not, I think there's not like 100% new ideas, but I think they do a really good job at looking at finding kind of doing microscopy and doing a lot of analysis to really establish that MyD88 forms these complexes, these kind of supramolecular complexes that are akin to small organelles at some point, which they call and apparently are called the middosomes, mydosomes, mydosomes. I'm not sure how to pronounce them. MYDDOSOME. And these mitosomes, they really are a structure that clusters together all the proteins that are related to signal transduction from toll-like receptors in one place. And these are dynamic structures that upon stimulation of toll-like receptors, you have these smaller structures that then kind of coalesce together and generate large mitosomes that then have a limited lifetime around, I think they say around 30 minutes, and then they kind of disaggregate again, and that's how the signaling kind of diffuses out. So in order to establish this, they kind of want to be very careful about not using exogenous kind of protein expression. Like they don't want to, they want to use the endogenous expression of mid 88. So they edit the genomes of human and mouse macrophages. These are cell lines, but they added a GFP protein so they can really see where in the cell this mid 88s molecules are. They also had some tags so they could do like they can just do pullouts from with tagged proteins, and they use this to really look at very closely at how are these clusters of MID88 changing throughout the activation. So the first thing they see, and they use something called internal reflection of fluorescence microscopy, is that indeed there's in a resting cell, you have small clusters of MID88, and upon stimulation, so they call them protomitosomes, and then they get activated and they kind of come together. They're in contact with the membrane, so presumably they've been activated by the toll-like receptors on the membrane. So they do this example with TLR4 that responds to LPS. And then they cluster in the cytoplasm, they coalesce together and make these large clusters that then disappear within 30 minutes. And this is really, they make a lot of points showing that this happens with different stimuli, with different toll-like receptors. And this is really not just like, these really organized structures that are formed. And they're very important for the signaling, and the formation of these structures is important for the signaling. And so the TLRs, they suggested that they are acting as kind of seeders. They seed the assembly of these myelosomes, and then the TLRs kind of decouple from the myelosomes, and it's just the MyoD88 that generates this cluster, which includes some of the other very well-known proteins, all these IRAC proteins, IRAC, I think 1, 2, 4, and then also these are part of the signaling downstream, and they together generate this large structure. Eventually, there's activation of autophagy, and it's through autophagy and autophagy-related pathways that the myelisome kind of becomes degraded after after it's done its job. So the point they want to make is that there is a organized structure, and they also look into microscopy, high resolution microscopy, like fluorescence microscopy, and they show that they form this kind of cylindrical structures, and that again they contain these proteins TREP6, IRAC2, IRAC4, and then particularly IRAC4 is important to kind of time the length that the myelosomes stay kind of assembled. So if you knock out IRAC4, then the myelosomes, they don't disassemble after the 30 minutes that are normal, but then they take longer. So as a consequence from some of the experiments that you do, they suggest that IRAC4 kind of accumulate, and at some point there's a critical amount of IRAC4 that then initiates the disaggregation of this whole cluster of proteins. So I think this is interesting because, yeah, I think nowadays that we're being a little bit more careful about understanding that location is so important, and kind of how the structures form. So we have this idea sometimes, or maybe it's just me, that have all this protein just moving around the cytoplasm, and it's just like interacting with whatever, and this kind of studies really show that there's a very orchestrated coalescence of these proteins, and this is very important. Like the formation of these clusters is important for the signaling downstream. They do some experiments in which they compare different cells that have different amounts of kind of these protomyelosomes. So they measure have like these small dots. So if they separate cells based on the amount of these dots that they have, they show that those that have the most, they're in kind of a super critical amount. So very easily, very quickly upon TLR stimulation, they initiate this organization, and this is kind of reduced when you have fewer, and this kind of, they suggest that this shows that you need this complex to form in order to have proper signaling.
So is the paper mostly about the complex then? Because I know
Yes.
MyD88's membrane associated. That's not new, and that it's not new that it's associated with TLR, but it sounds like it's the complex.
Exactly. So they see that it is initially, so it's not actually associated to the membrane, for what I understand. It is just before stimulation, it's found in these little kind of complexes, this little kind of oligomeric or monomeric, but upon stimulation, they get activated, they get seeded in the membrane by TLR stimulation.
Right. It's an adapter protein. When TLRs are activated, it goes to the TLR.
Exactly. So, and that activation it's like it feels like it's like a crystal. It kind of seeds these clusters, and then these clusters, they find each other and they coalesce into a larger, and then you have this large myelisome that you have maybe a handful of those in a cell, and you see them. They're like these big structures that you see with microscopy. Um, so I thought that was it's hard because I was like, yeah, for me it's like, oh, I might idea. It's like, okay, it's just there, but it's actually like pretty interesting that there is this separation and that they kind of I think they make a good point. Of course, the paper is very detailed and they do a lot of studies and they knock out different components of the mighty 88 of all these proteins because they do a pull down and so all these proteins are associated in these complexes and they knock them out. So I think in the end they made a good point showing that this seeding and this coalescence of these metasomes is important. It's like it's not just it's like a part of the function of how the signaling works. And because then you have all the components of the signaling in one place in this organized structure. There is some proteins are found like inside out, like there's like this also this spatial distribution of the proteins of the complex, and that doesn't seem to be just random, but there it kind of seems to be following some kind of order. I thought was quite interesting.
Super interesting. All right. Well, I don't have a good segue here other than I guess I'm doing something also on the cell membrane. But what I am doing explores adaptive immunity, and I'm not ashamed to admit it, Miss I, a preamble of innate immunity here. All right. So the title gives away the story. But that's not necessarily a bad thing. It is progesterone-driven B7H4 contributes to oncofetal immune tolerance. That's in Cell. The first authors, whose names will also be subsequently butchered, Jiali Yu and Yijuan Yan, and the last author is Weiping Zhou. And what came out on July 4th. So, B7H4 is a protein that is membrane bound and no one really knows what it does. But it has been theorized to be involved in the immune system. It interacts with CD8 cells, and they kind of know that already. But what they are looking at is immune checkpoint blockade. But instead of getting at it from the standard mechanisms, they're like, well, what is a system that is tolerogenic that allows something to grow in something without the immune system going gangbusters, even though you have a whole bunch of mismatched HLAs and everything else. Oh, pregnancy and that whole thing, right? So immune tolerance is a hallmark, and checkpoint activation is a hallmark of pregnancy, and understanding it could lead to insights into cancer. And so they do a screen early on and say, what's up in cancer that's also higher in pregnancy? Maybe that's a checkpoint molecule. Okay, so now we have B7H4. They call it an oncofetal immune checkpoint tolerance. Checkpoint. It is an oncofetal, which is new words put together for me.
Are you saying a baby's a tumor?
Yeah, close enough, I guess.
It's an adorable tumor. It's an adorable little parasite.
Fetus is a parasitic tumor. Yes. Okay. So to kind of keep things going.
Sorry, I just had to say, babies are awesome. I'm just, I don't want my listeners to think I have something against babies. They're beautiful. They're great. They're needed.
You're gonna get in trouble.
Any mothers in the audience, please don't hate me. Go.
All right. There you go. So at the maternal fetal interface, B7H4 is explained mainly in trophoblast cells. They look at this in humans, map it out, then map out the equivalent in mice so that you can actually do follow-on experiments, because they have human embryonic debris, so they can map the cells, but then you can't do any of the follow-up experiments in humans for very obvious reasons, because human fetuses are not tumors, Brenda. So that being said, they then do some crossing, right? So they do a knockout. and then they cross black. They do a knockout in versus bulb C mice, so that you have allogenic mating, and knock out. And then doing the knockout matings of the B7H4 leads to. So as expected, what happens is it's decreased in the female B7H4 knockouts, and if you do the mating, they have more fetus reabsorption or embryo reabsorption in pregnant mice, in pregnant B7H4 mice. So on the mother side, it matters, right? So the fetuses don't make it as often if you knock out the checkpoint. And guess what? They see changes in immune cell function, and natural killer cells are expressing more activation in these mice. And if you do it in a RAG system, a RAG knockout system, the effect goes away. So guess what? The adaptive immune system is also involved. Cool. That's the first part of the story. They then, and then they map out the immune cells and say, look, there's CD8 happening here. They look at the MHC1 complexes being expressed on trophoblasts and map that over and think it's really the trophoblast expressing B7H4 that's mitigating this potentially in combination with the MHC1 next door on the same cell. Okay. So then what they do is look at the fetus. So they take knockout mice and then cross those to heterozygotes knockouts, so that the fetuses are either plus minus or minus minus, right? And then the mom is minus minus. And minus minus fetuses do less well. In the uterus, they see more fetal reabsorption of the minus minus fetuses than the plus minus fetuses. If they do embryo transfer experiments, which you can do in a mouse, you see that putting the minus minus mice reabsorb more, even if you put it in a wild type host. So how is this happening, right? So they've basically figured out it's important on both sides, in the fetus and in the embryo, right, and in the mom. Progesterone promotes the expression in cancer and in the placenta. Okay, speeding along here. So they do that mapping. They do progesterone analogs. They do antagonists to reverse it. You see all the effects you'd expect by playing with the protein by drug. And it promotes breast cancer progression by inhibiting CD8 cells. So if you put progesterone in, you get more of this, and you have inhibition of CD8 cells that they show. Well, how is this happening? They map this down to the enhancer, doing a TAC-seq and similar work, and find out that it is, they use, they find it's a minus 58 kilobyte enhancer region upstream of B7H4, affecting by progesterone to drive this. So they map it down to where it's happening. And it does this by what's called the P300 BRD4 axis. The progesterone receptor pulls in the P300 PRD4 complex, and then that P300 is a histone acetyltransferase. And so progesterone pulls that to go upstream, it to do its acetyltransferase activity, and then that leads to the downstream enhancements. They don't just say, oh, it's bound in this promoter region. That's important. They also map out the protein axis involved in that promoter region. And BRD4 is the bromodomain containing 4 protein that's an epigenetic reader of H3K27 AC, right? So you have the P300 causes that to form, BRD4 binds, that is a epigenetic reader, and that then pushes the transcription from progesterone. So they map it all the way down. And then they show that in ovarian cancers, they go into mouse models and show mouse models of cancer that this, if you inhibit its production, you get a better outcome. So it's if you take a BRD4 degrader molecule, that especially alongside PD-1 checkpoint blockade, works in mouse tumor models, essentially.
Can you remind me, how is it exactly that B7H4 mediates?
They don't know.
Ah, okay.
It activates it is a checkpoint molecule, but they don't really know what it's doing. It's a cell surface molecule. It's a tolerogenic, and they don't know exactly what it's doing. A potential involvement of CD8 cells.
Okay.
It's on the surface. MHC seems to be important, so HLA seems to be important. Progesterone drives it, creates tolerance. There's worse outcomes, right, with cancer like this. Like, so, high levels of this in cancer is bad in people.
Mm-hmm. Yeah. Yeah. But then, do they hint or at a way of limiting the expression other than just removing progesterone?
Yeah, so they have a molecule, they have a degrader of BRD4, which is what they used.
And is there any chance that the BRD4 is not, is it specific for this? I assume BRD4 is probably involved in many things.
Yeah, it is not specific for this. It'd be a broader chemotherapy, but it is a degrader of a histone. So it's not going to be as broad as like, a topoisomerase inhibitor or doxorubicin
Mm.
or any of those
Hmm.
things, right? It degrades a epigenetic reader. So you're a little bit more narrow than screw up all the DNA of the cancer cell.
Right.
But that's true of checkpoint inhibitors anyway. It's not like PD-1 only exists in the cancer environment.
No, that's true. That's true. Yeah, but also no, but my point was also like probably there's other genes whose if expression could be affected by degrading that.
Yeah, absolutely.
All right. Okay, so why don't we end up with some interesting paper, like all the papers we present here, right? Um, so this one is called Nasal Tau Immunotherapy Clears Intercellular Tau Pathology and Improves Cognitive Functions in Age Tauopathy Mice. First author is Sagar Gaikwad from last author Rakesh Kayed from the University of Texas Medical Branch in Galveston, Texas. Um, I like how now, there's a lot of like this neuroimmunology, and I think it's great. So we are moving from treating a cancer with immunotherapy to treating so many other things with immunotherapy. And of course, neurodegenerative diseases that include, accumulation of pathogenic proteins are a great example of something that we should be able to target in a way. And so this paper, as I mentioned, so it looks into treating this accumulation of tau, toxic tau protein inside neurons, which is a hallmark of various neurodegenerative diseases, including Alzheimer's, but also, pig disease and um, other frontotemporal dementia. I don't know. There's a lot of other diseases in which cognitive decline is accompanied by accumulation of proteins such as tau. And tau, this is, I want to say because I think us, pure immunologists are not super familiar with how this works. But tau proteins, they is a microtubule associated protein that normally helps stabilize the microtubules in neurons. And this, is probably very important because neurons have this long axis. So probably they have a lot of microtubules keeping things in good shape. And one of the things that you observe in diseases like Alzheimer's is that there's this accumulation of tau that is abnormally phosphorylated and forms this kind of tangles, this aggregates inside neurons. So they're called neurofibrillary tangles or NFTs, and this disrupts the neurons, and as part of something that leads to neuronal dysfunctional death. There's also other things that happen during Alzheimer's disease. This is different to amyloid plaques. So this is a different type of protein. So these are separate processes. But well, so one of the ways of trying to get rid of this protein is by targeting with antibodies. So ideally, in principle, if you could pick up this protein and just cover it with antibodies, then it would get degraded. But then when you think about it, there's a bunch of complications. One of them is This is inside your brain. And of course, we do have this blood brain barrier, which we know is not as stringent as we thought, but it still limits the access of drugs that you put, in your blood system. And also the tau, this toxic phosphorylated tau, is inside your neuron. So how do we get this inside the neuron? And there has been some clinical efforts into using monoclonal antibodies that are targeting tau protein, but they're not really good at getting the tau protein that's inside the neuron. So it doesn't really help once you have an onset, once your neurons are already starting to accumulate. So basically, the risking just giving it all out in one go. This study, they're looking into finding they look into a particular clone that they have this monoclonal antibody that can specifically recognize tau, misfolded tau, that is toxic version of tau, and that the way that they are administering it through the nose and uh, by putting into mice cells, then that allows the antibody to get taken up into the neurons, and that actually really helps to uh, clear out tau, intracellular tau in their in vitro studies, and they do some mouse studies. So this antibody, they call it toxic tau conformation specific monoclonal antibody, TTCM2. And they show that this TTCM2 can recognize specifically this phosphorylated tau. So there's other antibodies that are recognizing phosphorylated tau. So they do a lot of studies in vitro models, and they show that indeed, and also like there's this mouse model that has that develops that has this human tau, and it's used as a model for Alzheimer's in mice. And so they use all this they study all this slide from brains from mice. I think they also have some slides from human people that unfortunately died of this disease, and they show that indeed this antibody can recognize this particular toxic tau, and this fibrils that it forms. And they also show that they can put it inside a micelle, so surrounded by, I don't know sure exactly how they make them, with some lipids, and by putting the monoclonal antibody with this inside these micelles, and they inject it, like this is added to the nose, so the nose has like very fast access to the brain, especially in mice. I think in humans as well. I don't know if it's so quickly also in humans, but then they see that they can indeed deliver this antibody into the brain of the mice by administering it in the nose. And by doing this, this antibody is taken up. So these mice cells allow the antibody to be taken up into the neurons. And then what's very interesting is that they actually see that these tau aggregates get cleared. And this is something that I didn't know so much, is that there is a cytosolic Fc receptor called TRIM21, which is apparently of broad reactivity to several antibody Fcs. And if this antibody goes into the cell and binds to the tau aggregates, then it can recruit TRIM21, and this initiates kind of degradation. I'm not sure if it's by autophagy, but then it degrades the tau, this tau complexes inside the neurons, and then it clears tau complexes inside neurons. So they show this. They do some experiments in mice, in these a human tau mice, and they see improvement in cognitive function. It does seem to protect the mice from the worst of the disease. And they show that TRIM21 is very important because they do some siRNA treatment of cells in vitro, and they show that then the clearance is reduced. So basically, they suggest that by doing this, you can give one round of immunotherapy with these antibodies in this in these mice cells is sufficient uh, to what would be significantly reducing cognitive decline in mice. So that's very interesting.
It is interesting. I would think the delivery mechanism is interesting, but the tau hypothesis is currently being very challenged in Alzheimer's, and like drugs on the market that
Yeah.
target tau are now being pulled. They were approved rapidly and then now being pulled, and they don't really work. And so I don't know if the mouse
Well,
model really capitulates human disease. I don't know if clearing tau actually treats Alzheimer's.
Well, I think that the point that they make, at least, and I'm not, I'm aware of those, but it wasn't like there was this FDA quick, like fast track, but it wasn't. I thought it was from amyloid, for beta amyloid, not for tau. But that was kind of problematic and was very expensive. And was like, do we really want to prove this? Is like the point that they make is that it doesn't. Once if you have already established disease, then the tau is inside the cell. So unless you can have a method of delivery that will get the antibody inside neurons, then it's not going to work. So they also they do make that point that they if you only add if you only administer the antibody, but it's not taken up inside the cell and in order to interact with intracellular tau, then they don't see this effect. It has to be delivered inside the neuron.
But my understanding is the drug was all based on the fact that it could go in the blood-brain barrier and remove tau from the neurons in the brain, and it still didn't work
Mm.
after it was approved. Got pulled recently. There's a whole big rigmarole about the whole thing.
Yeah.
But also the tau hypothesis may be true in this mouse system, but not true in people. Like tau is a
Yeah.
symptom, and many healthy people have lots of tau in their brains.
Yeah. And also like the cognitive assays that they do in these mice are, yeah, it's
Yeah.
not the same
But the
as the
delivery
person.
system, I think, is actually still highly
Right?
meritorious. Right.
I think that's very interesting. And then you can actually get the antibodies inside cells and then they get taken up. That's kind of cool. That I thought was really nice.
Yeah.
Yeah.
We're going to be talking about biosensors here and other ways to measure things in cells with Dr. Shana Kelley at the Chan Zuckerberg Biohub in just a moment. But before we get to that, activate, expand, and differentiate your cells with cytokines, chemokines, and growth factors from STEMCELL Technologies. These reagents are validated to ensure reproducibility across a variety of applications for immunology and stem cell research. Explore more at stemcell.com/cytokines.
For the second part of our podcast today, for the interview, we are joined by Dr. Shana Kelley. Dr. Kelley is president of the Chan Zuckerberg Biohub in Chicago, and she's Neena B. Schwartz Professor of Chemistry and Biomedical Engineering at Northwestern University. Thank you so much for joining us today, Dr. Kelley.
It's my pleasure. Thanks for having me.
Thank you for being here. All right. So, as the resident, I guess, biophysics nerd, you have a lot of really cool technology that you're using to explore immunology, and it's always fun seeing the tool belt expand, especially as it gets harder and harder to do things and learn more and more. And so I'm really curious about a lot of your technology and having you be able to describe it for some of our listeners. And I wanted to start with these reagentless biosensors that you were able to come up with and use. And so I was wondering if you could kind of describe them for the lay immunology audience and what the advantage of having it be reagentless is.
Sure. So we wanted to develop these sensors that were, as you correctly stated, reagentless, because the eventual goal was to implant them in living animals and maybe one day in the human body. And so if we want to put a sensor into something that's living, it has to really be standalone, right? We would put it on a microneedle, kind of like how a continuous glucose monitor works. And so to do that, we have to be reagentless. We can't be adding reagents to the sensor while it's implanted. So that was really a requirement that we identified going into the work. It was actually really hard to get something that satisfied that requirement, but we did it.
So I have to say, I am not as knowledgeable of biophysics as Jason is. So I see one of your last publications talking about biosensors, in which you have a biopendulum. Did I read that correctly? So how does that work, and what exactly in this particular, this is a publication from a few years ago. What is it that you're measuring, and what is this whole biopendulum supposed to work like?
Yeah, so the pendulum is the way that we read out molecular binding to the sensor. So the sensor it's a piece of DNA. It's something like an antibody that can bind a protein of interest. And then we basically watch it swing on an electrode surface. It's actually an upside down pendulum. So if you can think about the piece of DNA as kind of the arm of the pendulum, and then the antibody or whatever binds to a protein is the bob. And then we're watching it swing. And it turns out that it swings slower if a protein binds to the antibody, because that part of the pendulum gets bigger and it's dragging through solution. And so it slows down when binding happens. So this is really, it's kind of like a molecular motion detector. And we're just using the motion to tell us about binding.
This sounds very similar to the principles for tethered particle motion assays, if you're familiar with those, where you use the persistence length of DNA, where it's small enough, this is for the audience, where it's like a rigid rod, and so it'll wiggle. And then depending on how far something up is down of that, you have more wiggle, or the mass at the end of it. That's cool to turn that into a biosensor. I always did it in vitro with enzymes.
Yeah. The trick here is that we use electrochemistry to read out the motion of the pendulum. And that's what also makes it implantable, right? So the continuous glucose monitors, right, are electrochemical sensors. Ours are also electrochemical, so very much compatible with the form factor that you need, again, to put it into the human body.
And then are you the kind of things you measure? You said you can put an antibody. Is this specifically for measuring proteins, or can you measure other kinds of molecules, as long as you can have some kind of binder to it and they're big enough to drag or to generate this movement?
Correct. Yeah. So those are the two things we need. We need a good affinity reagent that's going to catch the molecule of interest, and we need it to be big enough that it gives us a change in drag.
I like it that you're binding it with DNA. So you're just using as kind of polymers. It doesn't matter what the DNA is coding for. It's biological, it's long, it's strong, and that's all you need.
Exactly. Yeah. It as stated, it's a rigid rod, and it's a really good one because it's nice and short, doesn't bend or anything, which is what we need to make the pendulum work.
I see that here you're using the idea of this drag of the yeah, there being some kind of drag that is part of your measurement. And I think it's interesting because this also um, is related to another technology that you have been working on a lot and you developed, and I particularly find really cool, which is a method for sorting cells using a different system, a system in which you have this magnetic drag. Basically, you can label cells with magnetic beads and their antibodies according to some marker that you want, and then you can use this to put them through this magnetic field with specific shapes that then the cells that have the most beats, they get dragged most. But maybe you could give us a quick overview of this a little bit more elegantly than me, and maybe tell you what is the motivation behind using such a device. And I know that you have some big plans with it, or some very interesting ideas with it.
Yeah. So the electrochemical sensors that we just talked about are good at finding rare molecules in samples or in living animals. The nanoparticle based approach that you just mentioned is really good at finding rare cells. The way it works is that we take magnetic nanoparticles, really small ones, we functionalize them with antibodies. That antibody then is specific for a protein on the surface of the cell, or they can actually go inside cells as well. And then the particles label the cells according to the abundance of a protein of interest. And then we have these microfluidic devices that basically sort the cells according to how many magnetic particles the cells are carrying. And you're right, it's also a drag force based effect. The fluid is flowing through the microfluidic device. There's magnets on the top and the bottom. And depending on how many magnetic particles are there, it causes the cells to drag because of that magnetic force. That drag force is being there's the fluidic motion of the solution, and then there's the magnetic force trying to get the cells to stay put. And we're kind of balancing those two forces in order to get the cells sorted according to levels of protein expression.
So I have to ask, I know you have a PhD in chemistry. Did it happen to be in like physical chemistry or fluid dynamics or something along those lines? Because it sounds like the carry through theme is microfluidics, drag, and then the ability to apply that to immunology. That's the first part of my question. I guess the second part is, if yes, how did you go from that to all this awesome immunology stuff?
Well, I'll answer the second part first. So I am a chemist, but I'm the measurement kind of chemist rather than the molecule making kind of chemist, right? Those are kind of the two flavors of chemists that you typically see. And when I did my PhD, I was actually studying electron transfer reactions through DNA and just watching how they were modulated by the structure of DNA. So very, very, basic research. But we discovered that the transfer of electrons through DNA, you can modulate it if you start mismatching bases and doing different things to the structure. And so that's how I got hooked on sensing and realized that it was, using electrochemistry was a really powerful way to develop sensors. And also just got fascinated with like the development of new technologies and how that happens. I was fortunate to be a founder of a startup company that was based on the PhD work I did. And it kind of showed me how you can do basic research, develop a new technology, and then have it spin out and make its way to a commercial product. So I kind of just ended up in this journey that then informed the research that I've done in my own lab, which has been very technology development focused.
So you've had quite an interesting and awesome career with these technologies you're going to apply, measuring immunology, and now you lead quite a institute with quite a name. How did your career path get you from, classic academia, sounds like some spin-off companies, now kind of also at an institute? How did that like evolve, I guess, to use the biology term for you?
Yeah. So I moved to a new university about 3 years ago. I had been up in Canada in Toronto for 15 years before that, moved to Northwestern just to kind of start a new chapter and to come back to the United States, which is where I'm from. And I really thought I was just going to be settling into a new university and getting my research group kind of moved and restarted for, the coming 5 to 10 years or who knows how long. And I got a call from our vice president of research one day saying, hey, there's this open call from the Chan Zuckerberg Initiative. They want universities to put together proposals to create new biohubs. And those of us in the life sciences know about the San Francisco Biohub, which was the original, and how exciting what they've been able to do there is. And so I kind of hopped on this planning committee. Northwestern joined forces with the University of Chicago, University of Illinois Urbana-Champaign, and we had this 24-member committee that included the VPs of research from each university. And we spent the coming kind of 3-6 months putting together a plan, trying to identify the science that we would focus on. And very excitingly, we found ourselves going from, I think, one of 58 applications to becoming a semifinalist and then kind of a semi-semifinalist and then one of the top three. And then we won this competition. And what came along with that was $250 million worth of funding from the Chan Zuckerberg Initiative that would allow us to create the Biohub, which is a freestanding Research Institute. And, on the planning committee, the science kind of turned into my area. And so I ended up kind of more towards the front of the way the project was being led. And I was the one who got the call that we were successful with our application. And the person that called me, Steve Quake, who's the head of science at CZI, said, "And we want you to be the president of this new biohub." So, that's a day that's going to definitely go down in my history as a really thrilling one, and the start of a really incredible adventure. So that's how it happened.
So to continue on this and like the mission of the Biohub and kind of your career with engineering, it's a little sales pitchy, but also like maybe relating it to some of your specific work. Where do you think are the areas where this engineering, chemistry kind of I kind of more quantitative, hard approach that some people are less comfortable with? Where do you think that really, where is the next wave of opportunity in immunology for that, right? We think, I think of PCR as one of the last things that came along that really changed the world. And then eventually, instead of being water baths, you had Sanger sequencing and Illumina machines. And so that happened, right? And we've seen the same story with microscopy, right? And then light microscopy and now EM. It's kind of big things. Where do you think's next in the terms of engineering and sensing? And what do you think that could, 10 years from now do for the field?
Yeah, so there's many frontiers, and these are areas that we're working in at the Biohub. I mean, we've come a long way in immunology and understanding the roles of different kinds of immune cells, and really understanding kind of what's taking place in the human body when things like inflammation are happening. But you're right that it hasn't been very quantitative. And if you bring chemists, engineers, people with that, different way of looking at problems, I think when you ask questions, you get slightly different answers, right? And I'm really excited about what we're doing to bring in engineering-based platforms to ask and answer questions so that we can really drill down even further into how the immune system works. And there are you talked about kind of genomics and DNA sequencing. We've seen a huge revolution, right, in RNA sequencing and single cell RNA sequencing that's allowed us to characterize every cell type in the human body, right? But now it's time to move over to the proteome, the metabolome, the lipidome, like all of these omics that will give us that next level of information. So we're really driving towards that at the Biohub. We're using a lot of mass spectrometry based omics to canvas the proteome and the metabolome. We're using really new types of sampling technologies so that we can kind of move across a piece of tissue and understand how in space and time inflammation is evolving at the molecular level. So I'm really excited about that as definitely a frontier area that we're poised to make a big impact.
And I think as the last question, going back to that magnetic sorting, I will insist a bit on it because I sort cells all the time. And this looks like very promising. And I know that you are using it for finding very rare populations, which is a huge issue when you're thinking of traditional sorting protocols, because then the sorting times are impossible. Then the efficiencies, if you have very few cells, then you actually like the sorting just doesn't work. Like you just end up, the recoveries are sometimes too small and not enough. Are these platforms, have you working to commercializing these platforms? Is something that people, because I've never actually heard of it until I was looking into your work, and I thought to myself, well, but this is actually it's a very smart idea. Why can't I use it for my own research? Where is that in the future?
Yeah, well, you hit the nail on the head. You definitely have an appreciation of why we did what we did and came up with this technology that allows us to find rare cells. And we've been deploying that to do a lot of different things. We've used the technology to identify rare immune cells in the blood. And these are immune cells that are actually tumor reactive that we can identify in cancer patients. So we had to have the ability to really find those cells that were like a needle in the haystack to be able to identify them in blood. We've taken that approach and spun it out into a new startup. It's a year and a half or so old. And so the startup is then pushing this forward as a new approach to cell therapy for solid tumors, but they're also taking the cell sorting approach, building an instrument around it,
Mm.
and commercializing it. So I hope you'll be able to get your hands on it at some point. It's looking nice. It's into a nice piece of automated instrumentation. Another thing that we've done is use the approach on CRISPR edited cells. So you can create large libraries of CRISPR edited cells, right, where every single gene in the genome is knocked out. And then you can kind of sort through those libraries of cells to find ones that have interesting phenotypes.
Hmm.
And we've used that approach to look for modulators of immune cells, genes that regulate how immune cells secrete things into the microenvironment. So that's been really interesting. That's work that we're carrying on at the Biohub to really get comprehensive around understanding how immune cells work.
Hmm.
So there's a lot of different directions that we've already taken it and will continue to take it because it really, you're absolutely right that once you can get at those very rare cells in a complicated sample, you can ask a lot of questions that you can't ask with a fax machine, right? Which is the typical way that people sort cells.
Yeah, and also from a more technical perspective, like sorting is hard. Like it's hard on cells. It's hard. Of course the instruments are big, and you need a huge investment, and then you can only run one sample at a time. I think that's one of the main motivations was the throughput. That although there are machines that nowadays can process millions of cells, but still, if you're really looking for a needle in a haystack, it might not be enough. So high throughput is ever more critical. I think when you want to study these very rare cells.
Yeah, completely agree.
So you've mentioned a couple times a couple of startups you've done, right? You said one early, and this is another one. For that postdoc or that junior faculty who came up with the next cool idea that they think really could have some commercial interest. Uh, Besides the adage that many people say of "No, you don't know a lot and ask for help," I guess what would you advise first steps to be if you have any of for people who may be listening, thinking that like where should they go? Is it the tech transfer office at the institution? Is it also in addition to institution talk to someone? Like what should people do if they have that light bulb go off in their work and really want to see it, not just in papers?
Yeah, great question. I think the key thing is to figure out what a truly compelling commercial application is of your technology or whatever it is. That's often the hard part. There are things that sound good on the surface, but when you really drill into them as commercial products or things that address large markets or things that are investable, the ideas can kind of fall apart. So that's always step one. And that can be done by tapping people in your network that may have kind of a business orientation or some experience to help you. Talking to in the life sciences, you want to talk to clinicians, understand their perspective, and then trying to find a few. I mean, we usually do our startups with venture capital investment. And so if you can find a few friendly venture capitalists that will tell you what's wrong with your idea, that can be a great way to really, punch holes in them that you can then kind of patch and come up with that really killer application for whatever you're doing. So that again, that's the hard part. And you could go to your tech transfer office. Somebody told me very early in my career, Shana, there's nobody better equipped to commercialize your technologies than you because you really care. You really care about it. It's not one file of hundreds that you're trying to manage. And I have found that to be very true, that you kind of have to take ownership. You have to be the one to drive. Eventually, you want to do a handoff to somebody else, ideally. But I have stepped into startups that we've done as CEO or CTO or CSO because I needed to be there. The right person wasn't there, but I have also handed off to some great CEOs who have then been able to take it into the next phase.
I guess in the end, yeah, nobody cares about the project as you do. That's although sometimes it's can be exhausting, right? If you if everything that has to be done by you, probably finding a good team as early as possible is just as important for success.
Absolutely.
So I think we're reaching the end of our conversation. But on the now that we're talking about a career and maybe not directly science, we like to finish our conversation with our guests, ask them a little bit something more personal, a question, and so we have we would like to know you a little bit beyond your research. So there's this, we have two questions that you need to fill the blanks on. Uh, Question number one, when I'm not conducting research, I am blank. So what was your side personality?
Well, I think I would have to say it's being a mom. I have two teenage sons at home, which is like more than enough to keep me busy in my spare time. They're awesome kids. They're really interested in science. They're a delight to be around, but they are teenagers, and you can spend all day at work as the president of the Chan Zuckerberg Biohub Chicago, and everybody treats you like you're pretty smart and whatever. But you go home to your teenagers, and you don't get that same treatment. It's quite different and humbling, but I love it. And I'm fortunate to be married to another researcher. He's an electrical engineer, also working in academia. So we have this wonderful family, and it's a joy, to spend my non-work time with this group of
Yeah,
folks.
for sure. Jason, you can take notes.
I'm married to another biochemist and toxicologist, and given our dinner table conversations, my son, when he was not that old, asked how enzyme kinetics worked because it's just what was happening at the dinner table. So I could only
Kept
imagine.
hearing it.
I can only imagine similar experiences at the dinner table when your kids just kind of get grow up in the science, and it's just part of everything. So yeah, that really resonates.
They're
I'm
not
quite at the
teenagers yet,
teenage time
though.
yet, but I'm still, they still look at me like I'm crazy.
Yeah. Well, when they become teenagers, you explain things to them, and then they tell you that you're wrong. That's the usual dynamic, but it's okay.
And for our second question, if I could have one superpower, it would be blank.
That's a tough question. There are a lot of superpowers I would like to have come in handy. I guess, the other member of our immediate family is my dog, Jonathan. He's a Labradoodle. He's very fluffy and cute, but he does things that really mystify me. And I really wish I could read his mind. It would make my life a lot easier if we didn't have to have these like silent conversations, and maybe I could read his mind and he could read mine. And then,
or just talk dog. Maybe
yeah,
you can do
exactly. Exactly. So I think that would be pretty cool to get into.
Little power. There you go.
Mhm.
Very well. I mean, that's kind of nice. I wish I could read my cat's mind.
Yeah.
Diego's entonces staring at the ether. I'm like, what are you looking at, dude? What is that? So interesting. I don't understand. In any case, well, that's been a very pleasurable conversation. Thank you so much for joining us. So today we had Dr. Shana Kelley from the Chan Zuckerberg Biohub in Chicago.
That brings us to the end of our show. Don't forget to subscribe to our newsletter at www.immunologypodcast.com to get the show notes, including an episode summary and links to all of the interview and Roundup papers. You can also reach out to us on X at @immunopodcast or via email at info@immunologypodcast.com with feedback or to suggest guests. See you next time.